Today we're going to discuss about the multiplication or life cycle of phages in virus in the diversity of living world. We have discussed about the viruses, living and non-living characters, milestone of viruses and classification of viruses in previous class. So now we're going to see the life cycle of phages. Phage means the bacteria. That is, the virus attacks the bacteria. It is called phage, bacteriophage. Here it is of two different types of life cycle you're going to see. That is lytic or virulent life cycle, lysogenic or avirulent life cycle. Lytic cycle. So as I said, that is lytic means destroy, virulent means kills the host. It destroys the host. It disintegrates the bacterial cell. So it can also called as killer type. Virulent means killer. Okay. So it completely destroys the host cell, and there are the following steps involved. First step is adsorption, penetration, synthesis, assembly and maturation. So first we will see it here. So the first step is adsorption. That is, the phage is attached to the bacterial cell through the basal plate and the tail fibers. So basal plate has the hooks and the tail fibers. With the help of it, it attaches and synthesizes the enzyme to destroy the cell wall. To destroy the cell wall. And the next step is penetration. So it destroys the cell wall with the help of enzymes. Okay, the cell wall of bacteria was destroyed, and the genetic material of the phage genetic material is inserted inside the bacterial cell. After the insertion of the genetic material of the phage inside the bacteria, inside the bacteria, the phage DNA starts multiplying. You can see the more number of phage DNA here. It starts multiplying in the host cell, that is inside the bacterial cell. So after the multiplication, the next step is assembly and maturation. So it starts forming the shape of the tadpole. So and it got matured and ready to assemble as you are standing for the prayer in the morning, in the morning assembly, like that they are assembled to destroy the cell wall. So they got fully matured here. After the maturity, what happened, that is overpopulation occurs in this phase. So due to overpopulation, they want to come out of the bacterial cell. So what happened means they destroy the cell wall of the bacteria. The entire cell is destroyed. The entire bacteria was killed. After killing the bacteria, what happened? The cell wall is destroyed and the phage particles are released. So now you understood, no? The bacterial cell is here. This one is the bacterial cell, but here the bacterial cell is destroyed. So virulent type means it kills the host cell. So now we can see line by line, you can understand. Adsorption. The phage particle interact with the cell wall of the host, that is E. coli. Here they use the Escherichia coli, E. coli bacteria. So the process involving in the recognition of phage to bacterium is called landing. That is, it is attaching, no? You can see here the bacteria, that is on bacteria, it got attached. The phage material is attached. So that is called landing. So like an aeroplane lands in the airport, no? Like that, the phage material is landing on the bacteria. So once it come in contact, the tail fibers injects on the bacterial. How the mosquito put injections to us. Like that, the tail fibers put an injection on the bacterial cell. And to anchor for support the tail fibers and the basal plate are in. So that is called pinning. So they are putting injection in the bacterial cell wall. No, that is called pinning. Now penetration. Penetration means the injection of the DNA particle, as I said first, no. So you can see the injection, that is the phage particle here. So this is the phage particle is entering into the cell of bacteria. So that is called transfection. Now the DNA particle, the empty, that is the, now what we called it as this one, that is empty. So empty DNA particle we called it as ghost. So that is the person without the in the body, if a person is dead, then a person is called ghost. No, like that the DNA particle enter into the bacterial cell, the phage material, that is the empty protein coat, is called ghost. Synthesis. So after that, what happened? They degenerates here in this phase, synthesis phase, the degeneration of bacterial cell, that is bacterial DNA, occurs. So after that, the what will synthesize? DNA replication of the phage material starts synthesizing its DNA particles. Now assembly and maturation. So the phage particles are assembled. So the process of creating a tadpole shaped structure, you can see here the tadpole shaped structures are formed inside the bacterial cell. That is called assembly, and that is called the maturation. After 20 minutes of infection, 300, that is after 20 minutes, you can see the 300 phage particles in a bacterial cell. So 20 minutes is enough to see a matured bacteria phages in the bacterial cell. So the last point is the release. So the release of phage particle from the host by destroying the host cell. So this one is the host cell is destroyed and the release of phage particles occurs. So these are the points. They may ask the lytic cycle in your exam. Lysogenic cycle. As soon as the same thing. So here also the first process takes place, adsorption, the same thing as lytic cycle. It attaches to the, that is landing on the bacterial cell. So with the help of basal plate and the tail fibers, and the enzyme is used to dissolve the cell wall, and pinning also takes place. The first stage is same both in lytic and lysogenic. After the second step is, you can see the penetration of the phage particle, DNA particle entering into the bacterial cell. So after that, what happens after entering the DNA in the bacterial cell? This DNA, that is the phage DNA, multiply along with the bacterial DNA. So it multiply along with the bacterial DNA. So that is called, you can see here the colored one, that is or light color, light blue color like substance you can see here, no, that is called that along with the bacterial DNA. So that is called prophage. So when the bacteria, that is undergo binary fission, the prophage it also multiplies. So along with the bacterial DNA it multiplies, and this life cycle is an avirulent. It does not kills the bacteria and it is useful one. Based on this only the rDNA technology is followed. This technique is using, that is this is a natural one, but in biotechnology they are using this technique in rDNA, recombinant DNA technology. So you can see, as soon as the phage injects its the linear DNA into the host cell, it becomes circular. So it is linear only, but the host DNA is circular, so it becomes circular. So by the recombination of the bacterial chromosomes. So the integrated that is phage DNA is now called as prophage. Okay. So virion. Virion, it is an infective virus particle and it cannot able to replicate. So it is an non-replicating outside the host cell. Inside the host cell, it can replicate. So they may ask this virion particle, there is particle seen in the viruses. So the first one is virion. So infective virus particle non-replicating outside the host cell. Virion, circular molecule that is made up of single stranded RNA, and it does not have capsid, that is no protein coat, and this was discovered by T.O. Diener in 1971. So it has low molecular weight. Think the virion of RNA has low molecular weight, and this causes in Citrus X orticus, potato spindle tuber disease in plants. These virioids are causing because it doesn't have any protein coat, only the single stranded RNA. Virusoids. So it was discovered by J.W. Randles and the co-workers in 1981. And it is very small circular RNAs. Okay, circular RNAs. But they are linked. The molecule, the viral RNA are linked with another RNA also. That is called virusoids. Prions. It was discovered by Stanley B. Prusiner in 1982. A proteinaceous, till the virusoids or virion, virioids, these and all, genetic material, DNA, nucleic acid infectious particle. But prions, this is important 2-mark question. They may ask in your exams. So prion is a proteinaceous. The protein part is infecting, causes disease. So that was discovered by Stanley B. Prusiner in 1982. The causative agents, that is the prion, is the fatal, and it causes it affects the central nervous system. Okay, humans and other animals. Okay, so cow, I had given so many examples are there, but I had given the mad cow disease. Okay, the virus which infects blue-green algae is called cyanophages. The virus attacking fungi is called mycoviruses or mycophages. So I had given some of the questions related to the topic. Define the following: prion, virion and virusoids. Explain the steps involved in the lytic cycle. You have to explain the adsorption, penetration, synthesis, maturation and assembly, and the release with the diagram. So what is a prophage? The DNA, the infected DNA particle, the intermediate DNA particle along with the host DNA multiplies, that is, it joins with the host DNA. That is called the prophage. So thank you for watching the video. If you have any doubts, post your doubts in the comment box. If you like the video, give a thumbs up, share and subscribe to Science Easy Tech channel.